explore it in details. Pero before that, we will try to understand ano ba yung fates ng pyruvate natin. Okay? So this is wrong. Remove that. Hindi ko siya nabura. Okay. So sa aerobic condition or yung so may tatlong pwedeng pagpuntahan si pyruvate. It will be either in aerobic condition to yung oxidative decarboxylation para mag-produce ka ng acetyl-CoA. Si acetyl-CoA, isa siyang precursor sa citric acid cycle or the other components pa ng metabolic pathway. Si citric acid cycle naman, yun nga, si acetyl-CoA para mag-produce ka ng carbon dioxide. Si carbon dioxide is a waste of the body, pero ito naman yung food ng mga plants, di ba? Alam natin yan for the life cycle. At yung iba pang energy producing component ng cells, yung electron transfer reaction, which we will explore din sa mga next topic natin, to produce water and ATP generation. Okay? While in anaerobic condition or fermentation, you produce lactic acid para dun sa mga vigorously contracting muscles. So yung pyruvate mo will produce lactate. Dito naman magpo-produce ka ng NAD+. So we will I will discuss that also then separately sa fermentation process. And yung ethanol formation, which is usually happens sa yeast. Okay? Ito yung mga sa mga bakery, sa mga brewers. Ayan. So this is the methodology that they use to produce ethyl acetate. Right? Okay, so for the glycolysis, before natin isa-isahin yung reactions involved, let us look ano yung may mali 30.05. Right. So yung overall equation na meron tayo for glycolysis, yun nga, involve glucose to produce 2-pyruvate kasi from use 6 carbon to produce 2,3-carbon containing compound, si pyruvate yun, that you use up NAD plus and NADH, and you also use up ADP and ATP. Okay? So yung net na meron tayo is production of this component. Malalaman natin yan later. And yung energy na involved dito is a very favorable since they are all negative. Okay? So magpo-proceed yung reaction na yan. And I think this is wrong. Wrong sa part. This should be right. So, yung any importance ng phosphorylated intermediates natin. So, okay. No export of phosphorylated compounds. Dahil ang mga phosphoryl group, hindi sila kayang lumabas sa blood barrier. So, nako-conserve yung phosphoryl group sa loob ng cell and makakontrol mo yung concentration sa loob. Conversion of metabolic energy in phosphate ethers, nako-conserve yung energy natin sa phosphate ethers kasi kapag nag-breakdown sila, you will produce energy. Okay? Binding energy of phosphate group, lower activation energy, and increase in specificity. Yung effect niya sa mga enzyme. Ayan. So it lowers the activation energy and increases specificity. Many glycolytic enzymes are specific for magnesium 2+ complex. So madalas natin makikita si magnesium 2+ sa enzymatic process sa loob ng cell kasi it is a good substrate for the para mag-proceed yung mga reactions. Okay? Alright, so we will now discuss ano yung mga reactions involved sa glycolysis. Okay? So sa glycolysis, una nating ginagawa, so you from glucose after breaking down the stored sugars mo or stored sucrose like starch, you can now have the monomer glucose. So ayan, kailangan niya we will be using most of the time yung ganitong formation, uh, cyclic siya. So ayan, yung glucose natin, remember, it is a hexose. So meron siyang 1, 2, 3, 4, 5, 6 carbon and 1, 2, 3, 4, 5 hydroxyl group. Okay? So meron tayong number dyan is 1, 2, 3, 4, 5, 6. Okay? So in our phosphorylation of glucose, ina-activate natin si glucose para mag-proceed siya ng reaction by attachment of phosphate group. So gumagamit tayo dito ng ATP. So yung reactive side ng glucose, which is yung number 6OH, it can undergo phosphorylation, mag-form ng phosphate ester with um, enzyme hexokinase. Ayan. So, I think familiar naman pa kayo on what kind of mga enzymes yung meron tayo like kinases, isomerases. Ayan. So, yung reaction na ito is not reversible. Okay, so hindi siya bumabalik dun sa glucose. Hindi siya magpo-proceed diyan kasi favorable yung reaction natin to formation of phosphate group dahil nakakapag-produce ka ng energy of negative 16.7 kilojoules per mole. As I have mentioned earlier, mabilis yung or madalas natin makikita si magnesium 2+ since it is a good substrate to form or to induce reactions sa ating uh, metabolic pathway. Also, commonly nang alam natin na ang ginagagamit for phosphorylation is ATP alone, but in reality, yung Mg2+, it neutralize yung mga reactive oxygen part ng ating ATP in order ma-ensure na mas maatake yung phosphate group natin. Okay? So si hexokinase, ito siya. So this is a example. Ito yung hexokinase. And upon addition of glucose in this active site, nagkakaroon ng closing para ma-ensure na may na malapit sa isa't isa yung ATP and yung glucose para ma-ensure yung reaction. Alright? So ano nangyayari sa phosphorylation? Paano na phosphorylate si glucose? So I am asking here yung reaction mechanism ni phosphoryl group. Any glucose to glucose 6-phosphate. Right? So ano nangyayari sa kanya? So you start with your glucose. Right? Glucose. OH here. OH here. OH here. OH here. CH2. OH. Alright? Alright. CH2. OH. Alright. So remember na ang mga pangyayari dito sa ating reactions involves enzyme. So sa isang enzyme, may mga available dyan na mga component like base or mga residue ng nucleic or protein, mga mga amino acid. Ayun. Meron din mga ibang amino acid that can give hydrogen or that can abstract hydrogen. hydrogen bond. So, you have to keep in mind na available sila once you use enzyme, may specific area sila para maging responsible sa formation of that formation ng target compound, right? So dito sa ating hexokinase, may mga involved tayong component. So paano nagkakaroon ng phosphorylation? Phosphorylation using saan yan? Mm. Okay, pagpasensya niyo yung ingay sa labas. Drawing ko lang yung ating ATP. O, P, O negative. P, ay mali. Then, okay? So, paano nangyayari yung ating reaction here? So, meron kang base. Si base is a good nucleophile. Attracted siya sa electrophile. Okay? Ito na. Okay. Okay. And, alin ang electrophile natin dito? Yan. Si oxygen natin is partial negative, si hydrogen natin is partial positive. So between these two, attracted si base dun um, sa ating hydrogen. So upon obstruction ni hydrogen, yung oxygen will be looking for a electrophile also. And yung available natin na electrophile will be coming from our ATP or adenosine triphosphate. So, ano yung atakihin niya dito sa group na ito? Remember na we are using magnesium 2 plus to neutralize this part. So, kapag ito ay partial negative, partial negative, partial negative, partial negative, partial negative, means yung ating phosphate group nito is a partial negative, partial positive. Partial positive. Okay? So, attracted si O dun sa phosphate natin. So, it can obstruct yung bond ni phosphate group and ma-release to. Ayan. Para punta siya dyan. You will be having na yung iyong phosphate group leaving yung ATP, adenosine diphosphate, and yung glucose mo, OH, 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 CH2O, PO3. Ayan. So, meron ka na nung compound mo na glucose 6-phosphate. 6-phosphate. So, dahil we have formed this glucose 6-phosphate, yung glucose natin ngayon is an activated compound na. So, yung phosphate na yan, it is a good way to activate nga yung compound natin. Okay? So, using hexokinase, we have produced from glucose para makapag-produce ng glucose 6. Okay? Next naman natin na gagawin is to isomerize yung glucose 6-phosphate in formation of fructose 6-phosphate. So yung itong part na ito is a reversible reaction. So it can either go either way. And yung free energy na meron tayo is 1.7 kilojoules per mole. We are using a specific enzyme. So, mapapansin nyo sa mga susunod na metabolic pathway, sa mga reactions natin, specific ang mga enzyme sa substrate na kanilang nire-react, convert or kinikleave. It will depend on the specific enzyme. And in this case, we will be using an isomerase. So isomerase, same lang na ang ginagawa niya is, kumbaga para siya nag-rearrange yung component nung iyong compound. Okay? So isomerization of glucose 6-phosphate coming from an aldose to produce a ketose. Okay? So, ano yung gagawin natin sa kanya para ma-produce natin siya? So, you start again with your glucose 6-phosphate. CH2 PO3 OH 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 OH. Right? So, remember na meron nga tayong enzyme. And yung enzyme na yon, compose siya ng maraming amino acid. And this amino acid, yung mga reactive site ng mga amino acid, are mainly involved dun sa formation or dun sa responsibility ng enzyme na yun. Okay? In this case, we have here a histidine residue. So, si histidine, and N plus. Histidine residue. So, si histidine, meron siyang reactive nitrogen. And meron din tayo ditong lysine. Reactive nitrogen ng lysine. Okay? So, si nitrogen, yung lone pair niya will desire to have a pair kay hydrogen. And ito, magpo-form ka ng carbonyl group. Then, ito mag-abstract to look for electrophilic hydrogen para rin masatisfy yung need nung nitrogen to have a neutral charge kasi positively charged si histidine residue. So, magkakaroon ka ng ring opening. Okay, so oxygen. H. And here, oxygen. PPP. Okay. Yan. Lagyan nyo lang yung ibang mali. OH. OH. CH2OH. Oxygen. Alright. And in the same, ano mangyayari naman sa kanya? Kailangan mag-form ng enolate. So, yung enolate na meron ka, para ma-form yung enolate, so kailangan mong ma-induce yung electron movement on this site. So, si oxygen, mapupunta yung negative charge kay oxygen. So, paano mo siya may induce? So, yun nga, as I've mentioned kanina, merong mga amino acid residue na meron doon sa enzyme that can help yung reaction. So, in this case, merong glutamic residue. Yung reactive oxygen niya will abstract yung hydrogen dito sa second carbon. It will go here, mag-form ka ng double bond, and mapumunta din yung negative sign. Yeah, right. So you will now have OH, CH2OH, OH, double bond O, negative. Okay. So dahil si oxygen ay in negative charge, gusto niyang hindi dapat na may charge si oxygen in this kind of setup. So ang gagawin niya, i-abstract niya si hydrogen ng C2. Okay? So kukunin niya to. Kunin niya to. This will give off this. Perform ka ng double bond. And yung in mo kanina will move para ma-provide naman kay no. Hindi siya pupunta kay oxygen kasi satisfied na si oxygen. So pupunta siya doon kay kukunin niya ulit yung H na kinuha ng glutamic residue. Right? So kukunin niya to para mabalik dun sa enzyme. Kasi di ba ang enzyme, hindi sila kailangan magbago for the reaction na tumutulong lang sila para mag-produce yung reaction. So, dapat mapanatili natin yung characteristic ng enzyme. And in this case, yung H na kinuha kanina ng glutamic residue will be also get by yung in alkene part ng ating ketose. Okay? So, ano mangyayari sa kanya? Okay. So, meron ka na ngayong na-form na ketone. CO. H, CH2OH, CH2, sorry, PO3 to. PO3, PO3. CH2O, PO3. OH here, and carbonyl here, OH, OH. Right? So, meron ka na ngayon na form na ketone. So, paano naman tayo magkakaroon ng ring closure? So, remember, yung dalawa natin kanina na amino acid residue, they will also be involved para naman sa ring closure. Involved sila kanina for ring opening, they will be also involved sa ring closure. Ano mangyayari? Kukuhanin, remember na meron tayong nitrogen dito with H, lysine, and yung histidine residue natin, right? It will be reactive sa paanong paraan. So, kukuhanin ni saan yan? Kung ano yung naging involved kanina ng mga parts will be also the one na involved dun sa ring closure. Okay. Kukuhanin niyo yung hydrogen dito. Since ito ay maging reactive din, attracted siya sa electrophilic. Yan, electrophilic carbon ng ating carbonyl. So ito naman, yung carbonyl dito will abstract yung hydrogen from our lysine residue to give off at ma-produce pa rin yung ating enzyme kanina. So ano mangyayari? Meron ka ng ring closure. You will now have your fructose CH2OH, oxygen dito, CH2OP, P3, O3P, OPO3. OPO3. Also, the other OH. OH here, OH here, and OH here. So now you have your fructose 6-phosphate. Okay? So, ganun lang yung ginawa natin. So, you will be using yung component ng enzyme to produce your ring opening, yung histidine and lysine residue natin to open the ring. Then, yung glutamic residue natin will be reactive to form your enolate ion. And the same glutamate residue will give off yung kinuha niya kaninang hydrogen para makapag-produce ka ng carbonyl. Then, since yung carbonyl is yung carbon niya is a good electrophile, is an electrophile, magre-react naman yung ating mga histidine kanina. Yeah, yung mga amino acid residue natin na involved sa ring opening to close naman yung ring. Okay? Forming a ketose, which is ating fructose 6-phosphate. Okay? Ano mangyayari naman sa kanya? Since meron ka ulit reactive na hydroxyl group, you can undergo ulit ng phosphorylation. Okay? So yung step 3 natin will involve another phosphorylation. So phosphoryl group transfer to fructose 6-phosphate transfer from fructose to fructose 6-phosphate to form fructose 1,6-bisphosphate. To form fructose 1,6-bisphosphate. Okay? So, ito na yung step natin which we are, ito na yung committed step natin. Kasi hindi ito reversible and unlike dun sa mga naunang na-produce na component, they can undergo ng ibang metabolic pathway. But for fructose 1,6-bisphosphate, ang goal lang niya is para mag-proceed yung glycolytic pathway, yung glycolysis natin. So it gives up negative 14.2 kilojoules per mole at gumagamit siya ng phosphofructokinase, another kinase specific for fructose. Kanina gumamit tayo ng hexokinase para kay glucose, and this time we'll be using fructose kinase. Specified din siya for this fructose 1 for fructose 6-phosphate. Okay? So yung activation niya is under low ATP or high ADP or AMP. Kapag mababa yung ATP, pag mababa yung ATP, activation under low ATP or high ATP yan. Tsaka siya magpo-produce. Tsaka siya mag-undergo. Yung site ng PFK1 is a site of regulation. Dito pwede mong kontrolin dito on how nako-control ng cell yung flow ng glycolytic pathway since this is a committed step. Okay? So, paano nangyayari yung phosphorylation? Okay, same with our ginawa kanina. You will using another ATP and like this, yung sa CH2 mo, sa number, sa 1 mo, magkakaroon ka ng phosphate. Okay? So, same lang yan. Ayan. So you now have your fructose 1,6 bisphosphate. Okay? We are using bisphosphate sa mga component na nag-a-appear ng twice pero sa magkaibang position. Pero yung kanina sa triphosphate naman, ano yun, gumagamit tayo ng tri kapag magkakatabi naman sila. Pero pag magkakalayo sila pero nasa same molecule, we use bis. Okay? Alright. Ayan. So hindi na natin i-write yung reaction ni phosphorylation since same lang din naman siya sa glucose to glucose 6-phosphate. Ngayon, magkakaroon naman tayo ng cleavage from 4,1,6 bisphosphate to produce glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay. Ay. Anong una natin gagawin? In order for us to have this two component. Anyone? Yung reaction natin will be involve aldolase. Anong nangyayari sa aldolase? Anong nangyayari sa aldol condensation? We will be using fructose 1,6 bisphosphate aldolase to break yung bond. So, may cleavage tayo na mangyayari. This is a reversible reaction since meron tayong positively charged na energy. Okay. Ano nangyayari sa kanya? So, you'll start with your fructose 6-phosphate, 1,6 bisphosphate. CH2OPO3 CH2OPO3 PO3 OH OH OH. Okay. So, ma-form ulit yung magkakaroon ka ng ring opening. So, you have to open this ring. Okay, nasan yun? CH2OPO3 PO3. Ayan. And ang involved dito is yung mga mga residue or mga amino acid residue dun sa enzyme mo. Okay? So, si oxygen is in a yan, yung ina ng enzyme natin. Not H+. Since siya ay electronegative, it is attracted to this hydrogen. Yan. Mali. CH2OPO3 plus OH CH2OPO3. Right. Then, yung mga residue mo sa enzyme, NH, since ito ay electronegative, attracted siya sa electrophile carbon to give off at masatisfy yung positively charged ni oxygen. So you will now have nasaan na siya? Lipat ko lang to. OH and enzyme. This is his enzyme. CH2OPO3 OH 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 CH2OPO3. Okay? Ano mangyari naman sa kanya? So si oxygen, ito ay may hydrogen. Ito pala nailagay dito. May hydrogen yan. So this is a positively charged nitrogen. So yung kanya namang lone pair will be, ang goal niya is mais naman yung um, positively charged nitrogen. So kukunin niya yung hydrogen from nitrogen and give it off here to produce, oh no, sige. Right. Ano mangyayari sa kanya? Magkakaroon tayo ng formation ng positively charged oxygen here and yung nitrogen natin. Okay? Question, meron ba kayong application that kaya nyong mag-collaborative writing? Like website where you can draw at the same time? May access ba kayo doon? So para, okay. Sige. May alam kasi ako, bit paper naman siya, pero I'm not sure kung allowed pa ba akong maggawa. Wait lang. Hmm. Ano to? Okay. May sinend akong link sa message. Ayan. So, try nyo nga siyang i-access. Ayan. Okay. Sige. Yan. Para meron tayong collaborative na writing. So, pwede ko kayong ma-ask kung ano yung reaction na pwede mangyari for our reaction.
Asan po, sir?
Nasa message. Dito sa Gmeet. I think you can change your name. User. Yan. Yan. Isang page na lang yung gamitin. Ha? Sorry? Mag-change name. Hindi ko alam kung paano siya sa phone. Pero puntahan niyo yung profile niyo. Tapos may makikita kayo diyang general, cursors, users. Yun yung nakikita ko sa akin eh. Ayan, may nakapag-change ng name. Si Panganiban. Yan. Okay.
Hindi po naglo-load sa phone ko.
Ano kaya meron? Yung iba. Yung iba na access nila. Isang ano lang, isang page na yung gamitin natin. Right. Okay. Nandun na ba lahat? 6 pa lang kayo. Ano yung gamit nyo? Browser lang ng phone? Or laptop din ba kayo?
Laptop po. CP po.
Oo. Okay. Okay, so hindi pala siya friendly for mobile phones. Ayan, may nakapag-draw sa inyo. Pero walo lang kayo dito eh. Hindi ba kayo dapat? Paano kaya? Kasi hindi rin kayo makapagsulat sa PowerPoint ko eh. Okay, sige. Maghanap ako ng available pa na whiteboard para magkaroon tayo ng collaborative write-ups in order for me to, may instruct pa kayo on how the reactions happen sa ating enzyme or sa ating metabolic pathway. Okay. 7. So, hindi pa rin enough. Alright. So, back to our synthesis of ito to. Hindi pa to. Ah. So, nagpo-proceed pa lang tayo para magkaroon kayo ng ah, magkaroon ng inolate, makapag-produce kayo ng aldol, magkaroon kayo ng enol, not enol, makapag-produce kayo ng imine para mag-induce ng enolization. So, yun yung goal natin for this reaction. Alright. So, na-produce natin si CH2OPO3 and enzyme H and OH2 plus OH OH ba't ako tabingin OH CH2OPO3. Okay? And dahil si H2O ay good living group, since ito ay water, para ma-induce yung pag-live ni water, yung lone pairs ni nitrogen will react again with your component to release yung water para magkaroon ka ng enamine. Double bond N, enzyme, CH2OPO3, OH, tapos OH, OH, CH2OPO3. Alright. Yung base mo kanina here will be the reactive component, not A. Let's see. To abstract yung hydrogen-oxygen bond to form a double bond, and this will induce yung movement ng electron to produce or to mapabalik yung electron mo to nitrogen. Okay? So, nagkaroon ka ng breakage ng bond here. Okay? So, ano yung na-produce mo? Meron kang CH2OPO3, OH, H, double bond O, and H. So, meron ka ng G3P or glyceraldehyde 3-phosphate. Meron ka pa at plus OH, H, H, double bond, C, and enzyme. CH2, H. CH2, CCH2OPO3. So, papasensya niyo yung drawing. Wait lang. Isin natin. CH2OPO3. Okay? So, ano mangyayari next dito is, goal mo naman is mapabalik mo yung enzyme mo in its original form, and also magkaroon ka nung DHAP, yung dihydroacetone phosphate. So, ano mangyayari sa kanya? So, yung lone pair mo pa rin ni nitrogen will form a double bond here, and mag-i-induce yung movement ng electron para yan, yung H+. Kunin niya to. To form C double bond N, CH2O PO3, COH, CH2OH. Okay? Then, upon addition of water, H2O, yung component mo dito ng enzyme, kanina you use yung enzyme para mag-produce ka ng double bond N, and release mo yung water, ito. Now, you will use again yung water para ma-release naman yung double bonds yan. So, pabalik lang yung mangyayari. So, mangyayari sa kanya, maproduce mo yung ketone dito. So, you will have double bond O here. Tapos, yung dalawa mong dihydroxyacetone phosphate, OPO3. Ayan. Sorry, sana nakita niyo yung kung paano siya nangyari. So, yung goal natin from bisphosphate to cleave ay ito. Ayan. So, yun nangyari sa kanya from here. Ulitin natin. So, from here, yung glucose-6-phosphate mo, nagkaroon siya ng ring opening to form your ketose. Then yung ketose, since si oxygen is your reactive part, like electronegative oxygen, it will abstract a hydrogen from a base para magkaroon ng electrophilic carbon also. And yung enzyme involved dun sa ating aldolase, ayan, will induce yung movement ng electron to satisfy yung positive charge ng oxygen. So you will have a nitrogen-carbon-oxygen bond. And since si nitrogen is a positively charged, dahil meron siyang 4 na bond, yung abstraction ni oxygen, yung lone pair ni oxygen dun sa hydrogen ni nitrogen para masatisfy yung charge ni nitrogen. Now you will have a good living group na water, and yung para matulungan ma-remove yung water na ito, yung electronegative lone pair ni nitrogen will make a double bond, tapos mag-live na si water, and para naman magkaroon ka ng cleavage, yung aldol will yung kinuhanan mo ng hydrogen kanina na base will abstract, kukunin niya ulit yung hydrogen pero sa ibang oxygen naman. This will induce a double bond formation of oxygen, and dahil itong side na ito or itong carbon-carbon is a mabilis siyang ma-break, baba yung bond strength niya, dahil na rin dun sa availability ng mga electronegative oxygen, it can be easily break, and it can induce movement of electron na kung saan magpo-form ka ng alkene group and an amine. So nagpo-form ka ng enamine. So na-form mo na yung G3-phosphate, which is the important component para mag-proceed yung glycolytic pathway. And yung enamine mo naman, you have to convert it to dihydroacetone phosphate. Yung yung inamin mo will yung involved dito is the same yung enzyme para mag-form ka ulit ng double bond C double bond N, and upon addition of water, which is kanina yung nag-abstract will also be involved in this reaction para magkaroon ng C double bond O. So, reminder nga na ang enzyme natin assist dun sa formation ng ating target component. Hindi sila kailangan mabago. Hindi sila, mababalik dapat sila sa original state nila in order for them to be used again sa mga further reactions. In this case, yung enzyme natin dito na si aldolase ay involved para yung ating 6-membered ring na fructose bisphosphate will be divided into 3 carbon containing compound. Okay? So natapos na natin yung step 4. So sa step 5 naman, kailangan natin i-convert si dihydrogen acetone phosphate to G3P. Anong ginagawa natin dyan? Isomerase lang naman siya. Okay? So sa isomerization, conversion of teaching. Guys, isomerate lang tayo ng ating component. So from DHAP to produce G3P, you need triose phosphate isomerase. So reversible din yung reaction niya since you are producing a 7.5 kilojoules per mole. You have after the reaction, you will be having a two indistinguishable derivatives ng glucose. So you have yung 3 carbons from 6 carbons ng glucose. Okay? And ito yung end ng preparatory phase. So, ano nangyayari sa isomerization? So, yung DHAP mo, CH2, OPO3, and COH, given na meron base dun sa enzyme, it will obstruct yung, magkakaroon ka ng formation of enol. So, pag bumaba yung electron mo, ito, ganap siya na free hydrogen. So, magkakaroon ka ng CH2, OPO3, OH, in, and COH, H. Okay? And paano siya H. So para magka-produce ka ng aldehyde, another base yung kailangan. Kunin niya tong hydrogen na to. Then mag-form ka ng oxygen dito. Then yung alkene group mo dito from the base kanina will abstract yung hydrogen base bond para mapabalik yung state ng base kanina from your enzyme. So you will have C double bond O H and OH CH2OPO3. So, meron ko na ulit, glyceraldehyde, 3-phosphate. Okay? So, ganun lang. Yung base dito kanina na nagkuha ng hydrogen will be the same base na involved sa mga next pa na conversion ng DHAP to G3P. Okay? So, ganun lang yung isomerization. Isomerization na nangyayari dun sa ating step 5. And this ends dun sa preparatory step. Ang goal natin for preparatory phase is to create G3P from glucose. So, next naman will be the payoff phase. So, you will oxidize yung G3P to create 1,3-bisphosphate glyceraldehyde. Pero instead of you using ATP, ang gagamitin mo is a phosphoric acid, which is an inorganic phosphate. Si inorganic phosphate will attach itself dun sa glyceraldehyde to form a uh, acyl anhydride, which is a very high energy para uh, high energy siya yung bisphosphate glycerate, and it will promote yung reaction forward ng ating glycolysis. So, nangyari lang naman is yung phosphate group mo will create an ester bond with the aldehyde. So, you will be forming this bisphosphoglycerate. Okay? Alright. Ang ginagamit natin ditong enzyme will be glyceraldehyde 3-phosphate dehydrogenase. So, reversible yung reaction producing 6.3 kilojoules per mole. Ano nangyayari sa kanya? So, your glyceraldehyde, CH2O, CH2O, PO3, double bond O, OH, H. Dun sa enzyme mo na ano enzyme ang gamit natin dito? Dehydrogenase. Meron ditong reactive site ng sulfur na residue siya ng cysteine that will initially create a hemiacetal or thiohemiacetal. So ito will abstract this. OH, CH2O, PO3, oxygen, sulfur, hydrogen, negative. Okay? Tapos, since yung oxygen nga is may negatively charge, hindi siya pwedeng magstay like that. So, mangyayari, yung magka-form ka ng double bond, and itong hydrogen mo dito will be accepted by an NAD+. Okay? So, yung mga nicotinamide adenosine diphosphate is a good acceptor ng mga free hydrogen. So, ito will attach itself doon. Ma-form mo yung hemiacetal or thiohemiacetal, OH, CH2O, PO3. Okay? Ano naman next na gagawin natin? So, kailangan, ang goal natin dito is magkaroon ng for PO negative. Yeah, PO negative. OH, O negative, O negative is a good nucleophile. And ang pwede niya ma-abstract ay attracted siya dun sa electrophilic carbon, and si cysteine residue will be cleaved from your thiohemiacetal. You will be producing yung iyong 1,3-bisphosphate glycerate. So, CO phosphate CHO phosphate OPO3 OH H H H. Okay? Yun lang. So, the same, yung sa enzyme mo na involved na sulfur will come back kung doon sa original form niya para magamit mo pa siya for the next oxidation of G3P to 1,3-PGP. So, nagawa na natin si 1,3-PGP. After nun, magkakaroon na tayo ng creation of ATP. So dito na natin makukuha yung mga ininvest natin kanina na ADP. And remember, sa every reaction natin dun sa payoff, times 2 yung nangyayari. Dahil dalawang pyruvate yung na-produce natin. No, not pyruvate. Dalawang glyceraldehyde phosphate yung na-produce natin. Ang goal naman natin dito sa payoff is mag-produce ng pyruvate. So your 1,3-bis-phosphoglycerate will transfer its phosphate group to ADP. So gumagamit tayo ng phosphoglycerate kinase and another substrate din natin si Mg2+ to produce yung 3-phosphoglycerate. So yung 3-phosphoglycerate tsaka yung number 6 kanina natin na formation, kapag pinagkupol mo sa kanila, so si 6 ay endogernic, meron siyang positive charge, and C7 naman ay exergonic. And adding this two reaction will give us an exergonic reaction which is favorable to proceed. Okay, kahit na sabihin natin reversible yung mga reactions natin kanina, favorable sa common denominator natin na 1,3-bis-phosphoglycerate na mag-proceed in production of 3-phosphoglycerate. Okay? So ano yung nangyayari sa kanya? Binibigay lang naman yung phosphate group dun sa ADP. Next is the conversion of 3PG to 2PG. So magkakaroon tayo ng formation or kumbaga para siyang isomerization of phosphate group from 3 carbon, from carbon 3 to carbon number 2. So ano nangyayari sa kanya? Initially, we will thought na this just moves here, pero in reality, involved dito ay ang mga initially phosphorylated amino residue. So dun sa enzyme na involved, so meron kang COO negative, COPO3, OH, H. Yan. So dun sa enzyme mo, meron kang histidine residue. Dalawang histidine residue siya. Histidine na phosphorylated, yung isa. O yung isa naman ay hindi. Okay. Nangyayari dito, in-abstract nung nitrogen, yung hydrogen mo dito, to have this phosphate group. Yan. So you have now COO negative, OPO3, COPO3, H. E-diphosphate intermediate. Okay, so meron ka ng nitrogen H+, histidine. Ano mangyayari sa kanya, yung nitrogen mo kanina dito na histidine will also be involved on abstraction of this component here. Okay? Mangyayari, si phosphate, kukunin niya, then yung oxygen dito will grab yung hydrogen. Okay? Lang. Parang gulo na. PO3. Kunin niya to. And kukunin niya yung wala. Bura na. Hydrogen. Niya kanina dito. Okay? And you will be producing your 2-phosphoglycerate. COH, CH2OH, COO negative. PO3. Okay. Next will be the dehydration of 2PG para ma-form mo yung phosphoenol pyruvate. So ang meron na naman dyan will be creation of uh, makapag-release ka ng water. Okay, so sa enolate, ayan, form ka lang ng double bond here para, wait, let that be. Is abstract yung hydrogen mo dito para magkaroon ng double bond here. And yung water mo will be attracted dun sa water. Sa H+. Okay? So you will release yung water at yan, meron ka ng enol pyruvate. Next will be here is yan, yung giving off mo ulit nung pyruvate. Okay? Nung pyruvate, nung phosphate group. So yung acceptor nung phosphate will another ADP to form yung the pyruvate. So tapos na natin yung glycolytic pathway and formation of from glucose to pyruvate. Okay? So formation of second ATP from phosphoryl group transfer from phosphoenol pyruvate to ADP. And you will be using pyruvate kinase with substrates using potassium or magnesium or manganese. Irreversible yung reaction na to kasi mas favorable na magkaroon ng ATP formation. Okay? So dito na na-store yung energy natin and yung upon breaking ng ATP, yun nga, pwede mo pa rin ulit mag-proceed yung mga next na reactions. Okay? So, substrate level phosphorylation, since irreversible, important site for regulation. Yun yung kahalagahan ng mga committed step ng mga glycolytic pathway, whereas you can control yung formation ng mga substrate or formation ng mga metabolites. And upon controlling the metabolites, you can now have a balance, equilibrated concentration of the components ng glycolytic ng iyong metabolism. At kapag may mga enzymes na missing dun sa mga pathway mo that can give abnormal conditions dun sa system, and malaki yung possibility na may mag-emerge na mga diseases or mga abnormalities dun sa system. Okay? So sa overall glycolytic pathway natin, we have used only one glucose and invested 2 ATP, 2 NAD+. But 4 AD to. 4 ADP and phosphate. This is But in return, nakagamit tayo ng naka-produce tayo ng dalawang pyruvate which will undergo naman sa iba't iba pang metabolic pathway, and you produce for ATP. So, from investment ng dalawa, nakapag-produce ka ng 4. And in net, meron kang dalawang ATP. Okay? So yung glycolytic pathway is a tight regulated pathway dahil merong dalawang pwedeng paggawa ng pyruvate, isang anaerobic condition, whereas yun nga yung ginawa natin kanina, that you have 2 ATP produced, but in aerobic condition that involves oxygen, maraming ATP yung kaya mong magawa. Okay, so it involves ATP consumption, NADH generation, and allosteric regulation of enzyme. Specifically, yung hexokinase, phosphofructokinase 1, and pyruvate kinase. Ito yung tatlong committed step whereas the cell can control yung formation of its specific metabolites para maiwasan yung overly concentrated or overly masyadong maraming component sa system. Kasi kapag sobrang dami ng isang component, it can induce naman ng ibang abnormality. Okay? It can also be regulated by hormone, namely yung glucagon, insulin, epinephrine, and changes in gene expressions for the enzyme. So may mga sites that can change yung uh, characteristic ng enzyme at the same time para rin makahelp sila dun sa other reaction pa. Okay? So familiar kayo sa insulin. So sa insulin ay component ng katawan natin para mag-breakdown ng glucose. And ayan, so involved kasi sa ating topic ng biochem ay yung mga effect ng metabolites once lack sila or sobrang dami or may missing enzyme ka or missing protein, may mga disease-related talaga na magka-come up. For example, si type 1 diabetes mellitus, ito yung abnormality where mababang B cells lang yung napoproduce ng pancreas and resulting siya sa insufficient uh, insulin that cannot break down yung masyadong that cannot break down yung glucose sa katawan. And kapag hindi na break down yung glucose sa katawan ng tama, pwede kang magkaroon ng hyperglycemia which having a high level of sugar in your blood. And kapag untreated na yung type 1 diabetes, it can produce keto acid which is a life-threatening condition, especially uh, sa brain. Okay? Ang intervention na ginagamit dyan is to inject insulin. Okay? Yan. So, questions sa glycolytic pathway. May questions ba kayo sa glycolysis? On how we produce yung glucose to pyruvate? How about the others? Wala pa naman. Okay. Next is the feeder pathway of glycolysis. So, anong nangyayari naman kung paano natin napoproduce yung, or paano tayo nagkakaroon ng glucose, okay? So, either, sabi nga natin kanina, it will come from starch or glycogen. So, it starts yung formation ng glucose sa mouth dahil meron tayong alpha amylase sa ating saliva. It initially breaks yung mga sugar component dun sa ating kinoconsume na diet into oligosaccharides or short polysaccharides. And upon entering the stomach, yung alpha amylase natin from our mouth, from our saliva, will be inactivated due to the low pH na meron dun sa stomach. And pero may other alpha amylase na involved. It comes from pancreas. So yung pancreatic alpha amylase mo will be released para mag-form ka ng maltose. Ay yung mga maltose mo, yung mga para mag-form yung mga oligosaccharides mo to be or yung mga polysaccharides mo will be breakdown to maltose, maltotriose, and limit dextrin, and it will be further breakdown to glucose. Okay? So meron ka ng glucose. While in cellulose, tayong mga yung mga human and other yung mga animals din na iba ay hindi nila kayang mag-digest ng cellulose. This comes from plant, and yung mga ruminant animals, they can they can break yung cellulose using symbiotic assistance from microorganisms. So merong mga microorganisms sa loob ng katawan ng ruminant animals that produce cellulase. Yung cellulase will now aid to break yung cellulose to glucose, and yung mga microorganisms na yun can assist and then we'll use yung glucose para makapag-produce pa ng propionate or lactose, which is their component. While yung glucose na na-breakdown can be a beneficial na dun sa ruminant animal. So, kaya siya tinatawag na symbiotic relationship. So, yung animal will eat yung mga cellulose containing diet, then yung i-break down nung cellulase creating bacteria or microorganism which give sa kanya ng component para sa kanyang cells. At the same time, yung mapaproduce na glucose from breakdown ng cellulose ng microorganism will be a source of fuel para dun sa ruminant animal. So, symbiotic assistant siya. So, ito yung nangyayari sa right. Ayan. So, your dietary glycogen or starch break by will be break by alpha amylase to produce yung mga glucose mo and other sugars para ma-produce mo si D-glucose. Yan. Meron din tayong pag-aaralan mamaya on how this other type of monosaccharide ay nagpa-participate sa glycolytic pathway. Okay? Ayan. So, they will be involved para ma-produce pa rin yung glyceraldehyde 3-phosphate. Ito naman yung mag-undergo nga to produce pyruvate. Okay? So, lahat ng mga component ng mga kinakain natin like carbs, yan mga yan, they will be degraded to create specific metabolites which will be a precursor for many metabolic pathways. Okay? Degradation of glycogen and starch by phosphorylation. So, ang nangyayari dito, yung glycogen is a polysaccharide. So, marami siyang monomeric ng sugar. And para ma-breakdown siya, nagkakaroon ka ng phosphorylation dun sa site ng glycosidic band to produce glucose 1,6-phosphate. Ito, mag-undergo siya ng phosphoglucomutase para ma-produce mo yung glucose 6-phosphate. Okay? Which will proceed dun sa glycolytic pathway. These are just examples lang nung kung paano natin napoproduce yung glucose or yung glucose 6-phosphate. Kasi nag-start lang tayo dun sa glycolytic pathway natin sa glucose. Pero ang question is, paano naman natin na-achieve yung glucose? And these are the examples kung paano natin na-achieve or na-obtain yung mga glucose na pinaprocess ng ating cells. Okay? Ayan. So as mentioned din kanina, yung ating nakakapag-intake tayo ng lactose. And yan, familiar kayo sa lactose intolerance. So sa lactose intolerance, meron kang lacking of enzyme lactase. Yung lactase yung responsible sa pag-breakdown ng lactose. And kapag hindi na be-break or kung walang lactase dun sa body, hindi kaya ma-break yung lactose and magti-trigger yung bacteria involved dun sa degradation ng other components ng mga waste natin, and it will be considered as toxic. I-convert ng bacteria yung lactose as toxic products. Yung toxic products na yan, it will be a signal for the body para magkaroon ng rapid reaction or ng drastic reaction which may involve abdominal cramps or worse, diarrhea. Okay? And yung intervention lang na ginagawa since walang cure for lactose intolerance is to avoid drinking lactose containing diet. So controlled diet lang talaga siya. Right? So yan, ganun yung maging sistema natin in this class. Like we will be discussing different pathways, different enzymes involved, and this is related topics kapag yung mga pathways na ito ay naglalak or hindi siya nagpo-proceed. All right? So another feeder pathway for our kung paano paano naman nakakapasok yung ibang monosaccharide sa glycolytic pathway. For example, si fructose. So si fructose very component yan ng mga prutas. So, si fructose, nag-receive siya ng phosphate from ATP. So, in mast cells, yan, using hexokinase, so magkakaroon ka ng fructose 6-phosphate. Yung fructose 6-phosphate na yan, it can undergo aldolase para makapag-produce ka ng glyceraldehyde and dihydroxyacetone phosphate. While in liver naman, yung fructose will receive ATP to form fructose 1-phosphate. Hindi, yan, fructose 1-phosphate. Right? Yung galactose naman, another kind of monosaccharide. So galactose will be converted to glucose 1-phosphate using galactokinase. May disease din na within galactosemia na hindi niya nagagawang i-convert yung galactose to glucose. Another monosaccharide is mannose. So yung mannose will be converted to mannose 6-phosphate using ATP. So hexokinase din siya. Basta ang involved ay isang hexose. Ayan. Yung kinase will be transfer of A group. So dito is phospho group. Yung mannose 6-phosphate naman will be converted to fructose 6-phosphate using phospho-mannose isomerase. So yun din, yung fructose 6-phosphate will undergo dun sa glycolytic pathway. So kailangan silang either ma-phosphorylated or ma-isomerase or ma-convert to its specific component ng glycolysis. Either fructose 6-phosphate or breakdown na agad to DHAP or G3P. Ito pala, meron pala ditong reaction on how galactose enter yung glycolytic pathway. So your galactose, upon addition of ATP and with the aid of substrate Mg2+, yung galactokinase, yung hexokinase mo, yan, phosphorylated si galactose and it will undergo uridyl transferase para magkaroon ka ng UDP galactose and it will undergo forward para ma-produce mo yung UDP glucose or yung, not UDP glucose. This is the fructose 6-phosphate. Ayan. So, ilan naman yung reactions niya. So, from phosphoryl group transfer, then magkakaroon ka ng isomerization, magkakaroon ng movement yung iyong ketone from this is from alpha to beta. Ayun, isomerase. Alright, question so far before tayo mag-proceed sa fermentation. May wala ata. Alright, so we will be discussing naman yung fate nung pyruvate. Ano nangyayari sa pyruvate after ma-produce siya. So, tinatawag natin dun ay yung fermentation process or yung anaerobic condition. So, sa aerobic condition, si pyruvate will undergo carboxylation to produce acetyl-CoA which will proceed to citric acid cycle. Pag-aaralan natin siya for the next topic. Pero sa mga anaerobic condition or hypoxic condition, it will either undergo ethanol formation or lactate formation. Okay, so sa hypoxic condition, nangyayari yon sa mga rigorously contracting muscle. Yung sa mga athletics, kaya they can be athletic kasi yung mabilis na creation of energy for the muscles. Sa mga solid tumors, lactic acid bacteria, and submerged plant tissue. Okay? So, dito sa mga hypoxic condition, you have the failure to regenerate NAD+ sa mga muscles, resulting to lack of electron acceptor for the oxidation of glyceraldehyde 3-phosphate. So, ang intervention dito is magkaroon ng generation of NAD+ through fermentation of lactate ethanol. Okay? So yung una is lactic acid fermentation. Ang nangyayari sa kanya, yung reduction of pyruvate to lactate. Yan. So nakakapag-produce ka ng NAD+. Si NAD+ is mahalaga siyang component for glycolytic pathway para maging hydrogen acceptor for the process ng glycolysis. But in fermentation, dahil nga walang oxygen consumption, and you just reuse yung NAD plus and NADH from glucose to pyruvate and yung formation ng lactate, wala kang net change ng NAD plus and NADH. So walang nagagamit, ay walang ano, nare-recycle lang yung NAD plus and NADH. And it uses ATP to produce yung lactate. Well, in uh, ethanol, ethanol, ethanol fermentation, ethanol fermentation, you use, dalawa to. Okay. So you produce, ito yung dalawang exergenic condition. Yung pyruvate decarboxylase. Una is pyruvate decarboxylate. So this is irreversible conversion of pyruvate to acetaldehyde. So you have pyruvate here and you will just release yung carbon dioxide. So ayan, pupunta siya dito. And ay, yan eh. Right. Mm-hmm. Wait lang. Okay. So mag-move yung negative charge mo dito sa carbon oxygen bond, and yung bond mo dito will look for a proton to produce acetaldehyde. Okay? And after formation of acetaldehyde, you will be required to use alcohol dehydrogenase to form na yung ethanol mo. So you will be hello? Sorry. Ito yung involved sa yeast para makapag-produce siya ng makapag-produce siya ng as ethanol. Okay? Okay. So yung acetaldehyde mo will be converted to ethanol using alcohol dehydrogenase, using NADH to produce NAD+. So kaya tayo nagkakaroon ng ganitong reaction ay dahil we are lacking of NAD+, a good proton acceptor for G3P formation dun sa body. So, kaya magkakaroon ng alternative way para mag-produce ng NAD+ yung body. So, sa human alcohol dehydrogenase, ginagamit ito ng liver to metabolize yung ethanol. So, if you're a drinker ng alcohol, ayun, so, mahalaga na nagko-function well yung alcohol dehydrogenase mo dun sa katawan. No net change in NAD plus or NADH concentration, and you also use yung dalawang ATP. Okay? Ito yung reaction niya. Ayan. So, involves alcohol dehydrogenase is your NADH. to receive yung to abstract yung hydrogen sa kanya, you'll be forming NADH and your ethanol. Okay. So, gumagamit din yung katawan natin ng thiamine or yung cells ng thiamine pyrophosphate which carries the active acetaldehyde group. Si PPP is a vitamin B1 derivative and lacking of vitamin B1 may result to have a disease called beriberi, yung accumulation ng body fluids which is swelling, pain, paralysis, and can lead to untimely, ultimately death. Okay? So ito naman is just cleavage of bonds adjacent to a carbonyl group. Where's that? Yan, si pyruvate mo, using TPP, you will have a abstraction from your carbon to yung carbonyl group mo. Ayan. Then, to proceed and the TPP ion attacks the carbonyl group, pyruvate, then you will have here para magkaroon ng decarboxylation. And upon decarboxylation, you will have this, so nakita niyo siya. Ay, okay. Malabo pala. Pero yan. Ayan. So, upon decarboxylation, um, si TPP will have this at nagkakaroon ng resonance stabilization to produce yung uh, uh, hydroxyethyl TPP to proceed naman na mag-produce ng acetaldehyde. So, ito lang ay other pathway para makapag-produce ng ethanol. Right? While in other fermentation process involved naman sa food, ayan, yung yogurt. So si yogurt, fermentation siya nung carbohydrate mo dun sa milk by Lactobacillus bulgaricus. Yung lactate mo, dahil dun sa low pH na meron dun sa milk, yung mga milk at dun sa liquid na meron ka, yung there is a possibility na mag-precipitate yung milk proteins which turns out yung yogurt mo. Yung sa Swiss cheese naman, yung fermentation nung milk using propion ni Bacterium frondreschii, yung propionic acid doon at yung carbon dioxide. Yung carbon dioxide is the responsible component kung bakit nagkakaroon ng holes dun sa mga cheese. Okay? Other fermented food are kimchi, soy sauce. Dahil sa low pH na meron doon sa mga fermented products, it prevents yung growth ng microorganism. Kaya nag-stay na matagal yung mga fermented products. Okay? While in industrial setting, yung fermentation will be from mga available products like corn, starch, para makapag-produce ka ng mga biofuel like bioethanol and other cleaning and hygienic for hygiene purposes and also starting material para sa mga other compounds na kailangan for the industry. It can also form different acetic acids, formic acid, different acetic acid, different carboxylic acid, formic acid, acetic acid, propionic acids, and so on and so forth. So yung fermentation is not only happens inside the cell para makapag-produce ng NAD+, once na-lock na yung body ng NAD+, but it is also applied for industrial purposes to produce naman mga important products or mga useful products. Okay? Alright. So sir, paano naman po kapag nagkulang sa glucose? So since ang cell natin is a very versatile uh, component of the body, kailangan niya ma-regulate yung concentration ng glucose at nung mga products na napoproduce niya. And once na kulang yung glucose, may pathway na susundin, yung glycolytic pathway, pabalik. So pwede kang gumamit ng iba't ibang component to produce yung iyong glucose. Okay? So, ang tawag doon is gluconeogenesis. So, gluconeogenesis converts pyruvate or other related 3 and 4 carbon compounds to glucose. So in animals, yung glucose is generated from lactate, from pyruvate, from glycerol. Yung glycerol is a stored fat. So familiar kayo sa mga triacyl glyceraldehyde, like mga TGA, yung mga fat. Ayan. Stored doon yung glycerol. Pag tinanggal mo yung fatty acid, you will have glycerol. And from glycerol, you can produce glucose. So kapag naubos na yung glucose natin sa katawan, tsaka mag-undergo ng production, tsaka magbabawas yung body ng fats na meron tayo to produce yung glucose. So also, yung amino acids can be a good precursors to produce glucose. Ito lang naman yung nangyayari kapag wala ng sugar sa katawan natin. Kapag wala na tayong stored glycogen or stored starch or sobrang baba na ng glucose content ng katawan natin. In liver, pag-aaralan din natin to for the next topics, it involves Cori cycle na nangyayari sa muscles or sa liver. Okay? Samantalang sa plant naman, yung stored fats and proteins will create sucrose. Ito pala yung general formation. Okay? So you have from lactate, you will produce pyruvate, which will undergo to citric acid cycle para ma-produce mo yung phosphoenol pyruvate. So yung movement natin is pataas. This is the anabolic pathway. So phosphoenol pyruvate to produce yung iyong glucose 6-phosphate, then yung glucose. Then yung glucose will start again to be degraded para naman masupplyan yung demand ng body. In microorganism, glucose generation from acetate, lactate, or propionate. So yung propionate, we have said kanina, yung galing sa mga cellulase, yung mga microorganism na kayang mag-breakdown ng cellulose. They use yung byproduct ng glucose formation propionate para naman magbigay or magkaroon ng other component ng cells ng microorganism na yun, and it can also be used to produce glucose. Okay? Ayan. So, sa plants, ayan, from CO2 naman yung sa plants to produce 3-phosphoglycerate which will participate in this formation of glucose 6-phosphate. Okay? So, sa animals, it uses yung stored na mga component like glycerol, pero yung sa plants naman, it uses yung waste ng body yung waste ng animals, which is CO2. Okay? Which we will discuss net sa mga later topics. Okay, ano yung pinagkaiba ni gluconeogenesis kay glycolysis? So, 7 of its enzymatic reaction ay parehas lang. May involve na from formation of from pyruvate to glucose and glucose to pyruvate. It involves 7 enzymatic reaction na parehas. Pero may 3 bypass reaction. Ito yung reaction na kung saan pinagkaiba nila. These are irreversible steps. Kailangan yon para hindi mag-fail yung formation ng glucose. So at the same time, yun yung mga part na kung saan committed step yung formation ng glucose to pyruvate, at the same time yung formation ng pyruvate to glucose. Okay? So may 3 yun. Mamaya, isa-isahin natin sila. So ito lang naman ito yung 3 yun. The glucose, the fructose phosphate, and the step 10, phosphoenol pyruvate to pyruvate. Okay? So pabalik. Una na bypass 1 will be the pyruvate to phosphoenol pyruvate. So, dalawang exergenic reactions yung kailangan natin, and this is a committed step, a reversible reaction siya. So, una is kinakarboxylase natin yung pyruvate using a bicarb. So, yung pyruvate reaction with bicarbonate and ATP, you will produce an oxaloacetate. Yung oxaloacetate na yan will undergo um, will react with guanosine triphosphate para ma-produce mo si phosphoenol pyruvate. Okay? So, yung unang step will be pyruvate carboxylase. You react it with carboxyl with bicarb. And after production of oxaloacetate, yung oxaloacetate will be reacted with GTP using phosphoenol pyruvate carboxylase to produce your PEP. Ano nangyari dito? Okay? So the overall reaction for this bypass one is pyruvate with bicarb ATP GTP to produce phosphoenol pyruvate, and you use up ATP and GDP. So this is a very exergenic reaction kasi as you can see you break yung phosphate group ng mga uh, adenosine phosphate or guanosine phosphate. Okay? Alternative paths naman din for formation of pyruvate to PEP is either in mitochondria or in cytosol kapag galing kang pyruvate. Pag pyruvate, you will use yung oxaloacetate to produce NADH to produce malate. Yung malate naman will use up NAD plus to produce oxaloacetate in return. Okay? From lactate, ayan, you generate NADH by oxidation of lactate. Wala ata dito. Well, yung other bypass naman will be involved in formation of fructose 6-phosphate. Ayan. Si FB phase 1 is a different enzyme. So yung pabalik natin kay 416 phosphate and 416 phosphate, it involves phosphorylation. So meron tayong diyang ginamit na kinase, while yung pabalik we use as phosphatase. So removal naman ng phosphate group. And very favorable yon since you create a negative 16.3 free energy. The same time, yung isa pang bypass na pinagkaiba nung glycolytic pathway to gluconeogenic pathway is the first step, yung formation ng glucose from G6 phosphate, glucose 6-phosphate. So gumagamit ka rin dito ng phosphatase. Pag phosphatase, it means you remove phosphate group. Okay? So you just react your G6 glucose 6-phosphate with water para ma-hydrolyze sila to form your glucose and the free phosphate group, which will be received naman ng ADP. Okay? Hindi siya ma-receive ng ADP. Magagamit siya for other phosphorylated na phosphorylated na reaction. Okay? So this is also a exergenic reaction since meron kang negatively charged na energy and favorable yung condition for reaction of this. Okay, so may tatlo tayong site kung saan para ma-ensure natin na magpa-proceed yung pyruvate to glucose. Those are sa step number 1, sa glucose 6-phosphate, step number 1, 1, 2, 1, 2, sa 2. And yung last part na sa pyruvate. Okay? So, ini-ensure na kailangan iba yung enzyme na involved to formation ng glucose compared dun sa formation ng pyruvate para maiwasan yung sobrang daming glucose or sobrang daming pyruvate. Right? Next is the pentose phosphate pathway. So sa pentose phosphate pathway naman, it will be either oxidative or non-oxidative phase. So oxidative phase, you use NADPH or formation of NADPH or the ribose 5-phosphate. We have said earlier na si NADPH, it is a good component to induce yung reaction din ng mga kailangan ng reduction. And samantalang yung sa pentose phosphate naman, sa pentose naman, na ribose 5-phosphate, it is a good component on production of RNA and DNA. It is also a good component on production of coenzyme A in rapidly dividing cells, especially on bone marrow, on skin. So si coenzyme A, we will tackle it more sa citric acid cycle, which ito gives activation, high activation energy para mag-proceed yung citric acid cycle. Sa non-oxidative phase naman, you just recycle yung ribose 5-phosphate to produce glucose 6-phosphate. So ito yung schematic diagram niya. So for oxidative phase, yung glucose 6-phosphate mo will be involved in production of NADPH that will be involved in many reduction reaction. Okay? And you will produce 6-phosphogluconate, which in turns out will produce again an NADPH para ma-produce mo yung ribulose 5-phosphate that can undergo formation of ribose 5-phosphate to produce your different nucleotides or specific, more importantly, your DNA. And it can, kapag yun nga, non-oxidative process, para ma-produce mo naman yung glucose, you just transalodase, use transalodase, magkakaroon ka ng transadolization to produce yung glucose 6-phosphate. Right. Ito lang ay yung detailed reaction nung kanina. Detailed ano lang. 6 pentose phosphatase. Detailed information lang nung kanina. Okay? Ayan. In here, sa non-oxidative phase, either gumagamit ka ng transketolase or transaldolase. Maalin sa kung ano yung available na component nung cells. Okay? So sa transketolase, you transfer of 2 carbon fragment from a ketose donor to an aldose acceptor using thiamine pyrophosphate or yung TPP as cofactor. Sa transalodase naman, you just transfer a 3 carbon fragment. For example, in here, para nagtatransfer lang sila ng carbon. So nagtatransfer ka lang ng 2 carbons. So from ketose donor, you will magre-receive yung aldose ng 2 carbons to produce this component. Ayan. Using TPP as a cofactor and transketolase as your enzyme. Okay? So this is a detailed detailed reaction kung paano nagkakaroon ng stabilization yung reaction ng TPP with the ketose and with aldose. Alright. Yan. Lastly, this will be the last slide na. Pentose phosphate pathway can be regulated the same way sa production ng NADPH. So when NADPH is forming faster than it is being used in biosynthesis, mag-create yan ng signal that will inhibit yung first enzyme involved sa pentose phosphate pathway, which can result to more stock of glucose 6-phosphate. So pag nagkaroon ng masyadong maraming NADPH sa cell, ang reason nga nun, kapag sobrang daming NADPH, it can induce a lot of reduction and oxidation reduction formation, and it is uh, it can clog yung formation ng other components. So magbibigay siya ng signal or magkakaroon siya ng reaction with the enzyme involved sa formation ng NADPH, and mabablock niya yung formation pa nung yung synthesis pa ay yung pathway for pentose phosphate pathway, para ma-stay na meron kang enough na glucose 6 phosphate to produce enough ATP. At kapag na-reach na ulit nung body or ng cell yung enough ATP na kailangan niya, uh, magkakaroon ulit ng signal na, sige, pwede na natin ulit gawin yung production of NADPH. Ayan. Ganun yung nangyayari sa mga regulation process sa lahat ng mga enzymatic um, metabolism, enzymatic reaction natin. Pwede silang maging site to control nga yung iyong reaction in order to avoid abnormalities sa body na possible na mag-cause ng disease or can induce other interaction. May mga too much production ng mga specific component that can induce formation of tumors, formation of cancer cell, or induction of other diseases, which is a very hard thing to manage kapag hindi mo na siya na-control. So ang nangyayari sa cell, yung cell, it is programmed to balance yung component ng body. So may mga specific naman na pathway na pwedeng i-regulate niya, or may specific component yung cells kung saan magbibigay siya ng signal para ma-regulate yung involved na pathway para doon. But may mga cases na yung cells hindi siya ganun ka-reactive sa mga ganung signal, kaya may mga nagpo-proceed pa rin ng mga abnormalities. May mga interventions na ginagawa like mga medical intervention or gene therapy. Pero that's medyo malayo na yun sa topic natin. Pero yun nga, ang goal lang talaga nung mga pathways natin is to create something precursor and to either to use up or to create energy using ATP or NADPH, and para rin maging starting material siya for other components ng cell. Like everything na meron sa cell and everything na meron sa body natin. From our DNA, from our like mga components ng skin, components ng eyes, yung cells natin, that comes from the precursors that we take as a diet. From the proteins, from the nucleic acids na kinakain natin, kanan, that will be involved in production ng mga components ng cell. Kaya nagtutuloy-tuloy yung life. So mahalaga nga yung proper diet actually sa for all of us in order to sustain yung need ng body natin for growth and development. Ayun, so questions. That ends yung slide. That ends our discussion for this afternoon. Is there any question or clarification? Yung biochemistry will only, mag-focus lang talaga actually tayo sa biochemistry in kung paano nangyayari. Like yung synthesis part, application ng organic chem sa synthesis part, and more of discussion on what happens dun sa cells natin. So, ayan. So, based up dun sa maraming information that will come from biochemistry. And especially, ang approach ngayon ng mga biochemist is to, kahit noon pa rin naman, is to resolve ano yung mga specific diseases and what causes them, and saan pwedeng magkaroon ng intervention para maiwasan yung mangyari yun. I-share ko sa inyo yung project na pinag-aaralan ko ngayon, which is also my thesis. We are discovering anti-cancer drugs para magkaroon ng medicinal drugs sa Pilipinas that will cure a lot of cancer cells. So may tatlo kaming cancer cells na pino-focus ngayon, which is breast cancer cell, ano pa nga yung iba? Breast cancer cell, colon cancer cell, tsaka liver cancer, which is a predominant cases sa Pilipinas. Especially tayo ay, yun nga, marami ring way of living na medyo nagko-cause to have cancer. And mahabang proseso yung cancer research, anti-cancer research. And mahalaga yung biochemistry part para malaman namin ano yung part ng cells or cancer cells yung pwede naming targetin in order for them not to survive or not to proliferate. So yun. So yung mga interested sa inyo class na mag-proceed to medicinal chemistry, biochemistry is a good foundation talaga para mas maintindihan niyo yung medicinal approach. Para mas maintindihan niyo yung medical field in chemistry approach kasi it all lies down dun sa chemical part nung cells. Like in this specific topic lang natin, simple pentose phosphate pathway or yung glycolytic pathway natin, maraming pwedeng mag-result na diseases. What more dun sa mga complex cells or complex uh, body of cells like that. So ayun. So I don't know kung ano yung interest nyo after graduating, pero one of the I think one of the best field then is to be involved sa medicinal chemistry. You can be either involved sa natural products like maghahanap kayo ng mga natural products component that can be used as bioactive. May mga bioactivity that can be used sa mga like diabetes, mga ganun, yung mga diseases na meron tayo na very alarming at this time. Meron din kaming research actually na kung saan naghahanap kami ng mga anti-COVID products, nag-create kami ng anti-COVID drugs. Sadly, parang patapos na ata yung, hindi naman sadly sa part na patapos na parang tuwang-tuwa ako na may COVID. Pero, yun nga, hoping na matapos na yung COVID. And yung project na ginagawa namin will be beneficial if ever na magkaroon ulit ng surge ng mga like this pandemic. Ayun, so, ayun, parang ano lang, parang I'm sharing lang sa inyo yung insights na kapag nasa medicinal field ka, I think kilala niyo, teacher niyo si, ano, di ba? Si Ma'am Bugya, si Darlene. Alam ko nasa ba siya ngayon? Nasa pharma ata siya ngayon eh.
Pharma. Sa
Pharma,
Hertz.
nasa pharma ata siya ngayon. So yun, medicinal field din yung approach niya. Kasi, ewan ko, parang it brings us closer dun sa direct application ng science to the community. Pero yung part ko kasi sa cancer research, sobrang layo pa namin in reality na ma-public siya or maging available siya sa market. Pero somehow, it give us Filipinos na hope na may pinoproject tayo na somehow, sana, may ma-produce kaming anti-cancer. Ayun, so that ends yung actually yung discussion. Napa-share lang ako on how important yung biochemistry, especially kapag nasa medicinal field ka. Pero kung hindi nyo naman type ang medicinal field, well, napaka-versatile ng chemistry course. And ayun, I am encouraging everyone din na mag-take kayo ng board exam kasi iba yung labanan ng may license pagdating sa industry field. Yeah, marami rin tayong mga graduates ng BS Chem na nasa industry. I can't remember all lang kung sino-sino na sila. Pero marami na yung mga nasa high position. Ganun. Pero wait lang, sige, may questions ba kayo? Or wala naman? How about yung iba? Kung may questions? Okay, go. Ay, uy, sa consultation na 'to, ah. Okay, pero okay lang din naman sa'kin. Go. Hindi ah, wait lang. Ay, ano na kayo 'no, third year na pala kasi kayo. Tsaka yeah. Kailan ang ano niyo, kailan ng thesis title defense niyo? Wala pa naman? So title pa lang kayo. Tapos ano kayo, by group kayo or solo?
Solo daw po.
Okay, good 'yan. Good yung solo para at least control niyo yung mga actually, yung research nyo will be limited dun sa availability ng lab. Unless makakapag-resource out kayo sa iba. Ang mga approaches ngayon sa research, if I'm not mistaken, ay mga more on industrial, ay hindi, mga waste management. Yung sa last, may mga student ako last time na naging consultant nila ako. Yung naging approach nila is creating a mga wait lang, mga activated carbon para sa filtration ng mga heavy metals, parang ganun. Though yung heavy metal kasi ay medyo pricey kapag you will do it in your own. May nakapag-lab na ba kayo? I mean, wait lang. Balita ko merong UV vis daw sa ano, sa
Meron na po, sir.
nagamit niyo na siya.
Hindi pa po.
Hindi pa po.
Pero possible naman na pwede niyo siya gamitin.
Opo.
Okay. Actually, marami kayong pwedeng magawa na sa UV vis. Pwede kayong mag-search ng mga colorimetric techniques. Yung isa akong familiar akong colorimetric technique is pwede mong ma-assess yung mga sugar component or sugar content ng mga something like that. Pwede kayong mag-analyze ng ano ba? Yan, sugar component, mga colorimetric techniques, ano pa ba? Pwede kayong magkaroon ng kinetic study, like magkakaroon kayo ng reaction of this component, tapos ang gagawin nyo, i-expose nyo. Wait lang. Masyadong marami pala yung idea na nasa isip ko. Pwede kayong mag-kinetic study, like magre-react kayo ng magkakaroon kayo ng reaction with a light sensitive component, tapos i-assess nyo siya in hourly basis kung kamusta yung rate of reaction, then magkakaroon kayo ng modification on that system, tapos i-analyze nyo yung rate of reaction. Ganon. So para siyang optimization technique ng isang reaction system, and i-base nyo yung rate of reaction dun sa kung gaano karami yung concentration nung ginagawa nyo component. Tapos pwede nyo siyang i-apply nyo siya for other medicinal purposes or other purposes na pwede nyo gawin. Sulat ko. Pero yan. Nakaka-consult kayo. Colorimetric techniques. Colorimetric techniques. Actually, ito yung undergrad ko nung college. Yung ginawa na, wala pa kaming UV vis nun. So, nag-resource out ako. Nag-outsource ako sa FPRDI sa may UP. So, gumamit lang ako ng UV vis doon. In-analyze ko yung on how good yung prepared kong adsorbent against methylene blue contamination. So, in-analyze ko lang yung kung ano yung effect nung concentration, effect ng temperature. Ano ba ang effect ng temperature? Not temperature. Like, yung temperature, not temperature. Absorbent, amount ng adsorbent, concentration nung methylene blue, tapos time exposure, ganun. So you will analyze it ng ganun. Pwede rin kayong, yun nga, yung colorimetric techniques that involves analysis ng, ano bang mga color sensitive? May nagawa kasi ako dati like analysis ng sugar content. Sa sugar content kasi, pwede mong malaman yung GI. Actually, I actually do this sa ERI nung ano ko, nung on the OJT ko. So we analyze yung sugar component ng mga palay or yung mga bigas ng nasa ERI para maintindihan yung glycemic index nila. So I think baka pwede yung maging topic is alamin niyo yung sugar component ng mga commercially available ng mga rice. I don't know. Baka pwede niyo pa siyang gawin. Basta colorimetric kung gagamitin niyo yung UV base at i-maximize niyo siya. Pwede sa salt. Wait lang. Salt content. Hindi kasi ah, salt content. Pag salt content, hindi masyadong malawak ang salt. Specifically, sodium chloride ba to? Or sodium component. Kasi pag ganan, pag mga specific na mga heavy metal, ay mga metal, may mga, hindi siya colorimetric, mga, ah, yung mga, gumagamit ka na ng diode, mga electrode para i-analyze siya since mga ions na yung ina-analyze mo. Kailan ang due date nyo sa title? Wala pa. Ha? Baka maaga pa kasi. So, baka yung time nung mga first weeks or first month ay for you to look na sa mga research interest niyo. I don't know kung may possibility ba na bigyan kami ng chance to at least handle students like you. Pero kung mabibigyan man, edi that will be better then. And willing naman ako mag-assist sa inyo for that kind. At the same time, baka pwede nyong i-maximize yung LSP Los Baños. Familiar ba kayo sa LSP Los Baños? Ang LSP Los Baños, meron silang HPLC. HPLC, liquid chromatography. Sa liquid chromatography, pwede kayong maraming gawin. Like, sugar din yung meron sa kanila. Sugar analysis. Ito naman, pwede nyo, ang ginagawa kasi nilang ng Los Baños, isi-share ko na lang din kasi doon ako din nag-work dati. Uh, inaalam nila kung kung ano yung mga sugar na napoproduce after breakdown nung prebiotic or nung halaman after siyang ma-digest nung lactic acid bacteria na meron sila. So inaalam nila ano yung mga sugar component kasi pag sa HPLC kasi liquid chromatography, you separate yung sugar component niya. So malalaman mo kung may glucose ba siya or may fructose ba siya na na-produce and gaano karami yung glucose and fructose na produce, parang ganun. Try nyong makipag-coordinate sa Los Baños. Hindi ko alam kung nagana na yung AAS nila. May AAS sila, tapos ito pwede nyong pang-analyze ng heavy metal para sa cadmium analysis, sa chromium analysis, sa lead. Hindi ko lang sure kung nagana pa rin to. Tapos, ito will be a good research din kasi you can search for soil, yung mga component ng soil around Santa Cruz, parang ganon, o how good ba yung soil as taniman ng mga gulay, parang ganon. So malawak yung pwede yung mangyari. Pero kung magre-resource out naman kayo kung kaya nyo talaga, like kung may naman kayo, may cash naman kayo, well, pwede na kayong lumapit sa ASL, sa Analytical Science Lab ng UPLB, then ask them kung ano yung rate nung mga synthesis nila. Ayun. Yun lang. Sure. Laban guys. So medyo ano yan, medyo well, nakakapagod yung pag-iisip ng title. Tapos solo pa kayo. Well, yung time din naman namin, solo rin kami. Pero kaya niyan. Sino ang advisor niyo dito? Ma'am Joy?
Ma'am Ray.
Ma'am Reyes? Ay, hindi ko siya kilala. Okay. Ayun. So, sana may nabigay akong insights. Pero kung available nga na yung UV-Vis nyo, hanap kayo ng mga colorimetric techniques. Ay mga research na gumagamit ng colorimetric techniques. Kung UV ba to or UV? Ano, familiar, alam nyo kung UV, kung ultraviolet ba siya or visible lang? Or hindi nyo pa siya talaga, hindi nyo pa talaga nakikita yung equipment na. Okay. Okay, ayun. Si Miss Darlene naman yung instructor nyo sa Anachem 2, no? Anachem 3, tama ba? Instrumentation ba ang Anachem 3? Ayun, I think, if I can remember it right, na nag-apply ata si Miss Bugya for face-to-face class nyo. So, baka doon naman, ma-maximize nyo yung laboratory and yun nga, magkaroon kayo idea on how you can use yung UVS. Ayun. Okay. So, kung wala kayong question na, that ends our discussion. Or may questions pa ba? Gusto pa kayong i-ask? Wala na. Yan ko pala muna ito. Oo. Ano naman to, nasa PowerPoint naman siya. So, hindi naman siya nabura kanina. So, I hope lang din, ma-follow through nyo siya ng maayos. Ayan. So, nakasave na, isa-save ko lang naman siya. Then, okay. Yun lang naman. So, kung wala nang kayong question, pwede natin i-end na to. Then, we will meet again next Sunday. 10. Ayan. Para naman dun sa, anong next topic natin? So, yan. For our next topics.